Good afternoon. I'm sorry for the delay, but I welcome you on behalf of the Massachusetts Breast Cancer Coalition to today's webinar, Does Carcinogenesis Start in the Womb, Endocrine Disruptors, and Breast Cancer? My name is Michelle Dixon, and I am the Program and Development Director for the Massachusetts Breast Cancer Coalition, and I thank you for joining us today for this important discussion. I would like to introduce our speaker, Dr. Anna Soto. Dr. Soto is a professor at the Department of Integrative Physiology and Pathobiology at Tufts University School of Medicine and a member of the Centre Cavaillé at the École Normale Supérieure in Paris. Dr. Soto co-authored the book, The Society of Cells, in which she proposed the tissue organization field theory of carcinogenesis, in which cancer is viewed as development gone awry. Additionally, Dr. Soto has served on numerous government-sponsored advisory committees dealing with public health issues and testified before the legislative bodies. Dr. Soto, we welcome you.
Thank you, Michelle, and I also thank you, Cheryl Osimo, because the two of you invited me to talk about my research here. And this is very important to me because I still remember how we worked together with the Massachusetts Breast Cancer Coalition in the early 90s. So, in order to get started, I think I'm going to give you a very short refresher on endocrinology. So, endocrinology in a nutshell is the first part of my seminar, and that you must have heard it a zillion times. But we have to have it present. I mean, we have an endocrine system that is made up of many organs that produce chemicals called hormones. Those hormones travel in the bloodstream and they reach every organ, every cell in the organism, but they only act in what we call a target cell. That is a cell that has receptors for that specific hormone. And this is somehow I lost one of the, I don't know what's going on. Now I don't have control over my, okay. So it should be interesting to you that those hormones are present in the plasma membrane, for example, of cells, in which case, I don't know if you see my little arrow here, the hormone doesn't get inside the cell, but it triggers a very complicated mechanism by which the action, I mean the signal of the hormone gets to the nucleus where action takes place. That is one form. And another form of action is through receptors that are present in the nucleus, and that is for hormones like our estrogens that are soluble and can get into the cell directly. So be it as it may, only cells that have those receptors will respond. And the functions of the endocrine system are to maintain the consistency of and constancy of the internal milieu so that our cells swim in the appropriate condition of nutrients and pH, etc. They also regulate the nutrient supply to all cells of the body. And finally, they regulate development, growth, and reproduction. And this is the point in which, in this particular seminar, we are interested because we are looking at how these hormones control actually the development of many organs. Among the effects of hormones, we have to consider two. The activational effects that are reversible. So if the hormone is given, it produces an effect, and then when you remove the hormone, the effect disappears. That reversibility is exemplified, for example, by the menstrual cycle. And there are organizational effects that for the most part are irreversible. And for example, this is the effect of thyroid hormone on fetal and newborn brain development, and we are going to see a very sad example in a second. That is the case of congenital thyroid deficit. Here we have a boy. Its age is 22 years. but he looks like a young child. And this is because it was born with a deficit of thyroid hormone. And that happened, that created a problem, mental retardation. And it can be corrected when the fetus receives thyroid hormone, but later on is irreversible. So there you have an example of what may happen when we interfere with the control by hormones of development. In the next slide, instead, we see deprivation of thyroid hormone during adulthood, and this is a case, it's called myxedema, where you see like the tongue is swollen and you see the imprint of the teeth, and the skin is very dry and the hair is dry and people feel cold, but you give thyroid hormone and everything gets back to normal. So right there you have the essence of what we are playing with when we put chemicals in the environment that can mimic or interfere with the action of hormones. So now we are prepared to the second part, that is from natural hormones to endocrine disruptors. And I would start with a very personal story. That is something that happened here at Tufts to Dr. Sonnenschein's and my laboratory in '89. So at that time we were studying control of cell proliferation in breast cancer cells. So we had it, and the classical experiment is you have cells to which you don't give hormone and they don't proliferate, and then cells to which you give estradiol and they proliferate. So you see a big difference in the cell number between your control and your estrogen treated. However, we started seeing absolutely no difference. And so that smelled like a estrogen contamination. Where was this estrogen contamination coming from? And after 4 months of work, we found that it was in the plastic tubes where we store components of the tissue culture medium. We discovered that the estrogen was nonylphenol, a chemical that is used as an antioxidant in that plastic tube where we found it, and to make detergents and spermicides. At the point that we saw that it's used as spermicide, immediately we realized that people are being exposed. Nonylphenol was then the first xenoestrogen, that is environmental estrogen, found in plastic. And that produced a consequence that is very important to us. That was that we were invited to the Windspread conference. That is where the term endocrine disruptor was coined, and I was one of the 20 people at that conference. And this laboratory accident was used as the basis to elaborate the screen assay. That is a system to determine whether a chemical is estrogenic or not. And with that, we identified additional estrogens among pesticides, industrial chemicals, and other chemicals present in the environment. And that list appeared early on, to the point that it called the interest of the Massachusetts Breast Cancer Coalition, that presented us with the Marla Praising Award in '95 for our work and dedication in fighting the epidemic of breast cancer. That is another reason why I'm happy to be here, because we started the fight together. I mean, my fight and your fight. So we get to the definition of endocrine disruptors, which the most, I think the best definition is the one that the Endocrine Society coined, and I'm one of the authors together with Tom Soller and other members of the Endocrine Society. Endocrine disruptors are exogenous chemicals or mixture of chemicals that interfere with any aspect of hormone action. By that we mean it could be something that mimics the action of a hormone. It could also be something that inhibits the action of a hormone. It could be something that inhibits the synthesis of the hormone, or that impinges on the degradation of the hormone. Be it as it may, it alters the level of the hormones in our organism, with the consequences that we will see soon. Endocrine disruptors are present in objects, everyday objects that we have at hand, like the lining of tin cans, and I guess you are familiar with the intervention experiment of Silent Spring Institute, where we see that if we stop eating things that come from cans, our levels of bisphenol A go down, for example. But it's present in cosmetics. I mean, this is a very widespread problem. And I should say that there has been some action in Europe, but I don't see much of an action happening here, and it's a pity because this thing started in the United States. But, for example, there is a law now in France that prohibits the presence of bisphenol A and phthalates in food. So now we get to the probably the core of the presentation, and I have to introduce the fragile fetus. That is how we define, I mean, this is a term coined by Howard Byrne, one comparative endocrinologist that was present at Windspread. And the idea is that the fetus, and here we have a zygote, when we are just one cell, and we divide, and then we form this recognizable human cute baby. The fetus is very fragile because organs are formed. You see here it's just one cell, a bunch of cells, and then organs appear. And that could be altered by exposure to these chemicals. So the first case that we have that was very well known at the time of Windspread was exposure of diethylestylbestrol that was given to women to prevent abortion, miscarriage, and premature labor. But that wasn't true. I mean, it really didn't do any of that. People just thought that because women are full of estrogens, a little bit more estrogen would only be good. And it was given to millions of women. And this practice was stopped in America in 1971, when here at Mass General Hospital, Herbst and other physicians found that there was 6 cases of clear cell carcinoma of the vagina in young women, and they could trace it to exposure in utero to bisphenol A, diethylestylbestrol. And that was discovered only because clear cell carcinoma of the vagina is a very, very unusual cancer that only appears in old women. So that is how they got to understand what was going on. But that wasn't the only thing that happened with diethylestylbestrol, which is in any case a cousin of bisphenol A, the estrogen that we have that is ubiquitous in our environment. So what exposure in utero to diethylestylbestrol produce is something that we call the fetal xenoestrogen syndrome. A syndrome is a set of signs and symptoms that go together, and so that therefore characterize a given pathology because they have a common cause. And here we have the list of what happens when you are exposed in utero to these chemicals. This is women exposed before the 13th week of gestation develop clear cell carcinoma of the vagina. Additional anomalies are malformations of the oviduct and uterus, decreased reproductive success, increased risk of breast cancer at the age of prevalence, so that is much later, 20 years later, it was found out that women exposed in utero and even their mothers exposed during pregnancy had an increased incidence of breast cancer. And I should say that also there seems to be early cessation of menstruation and early appearance of menopause in these women. So now we get to the real center of our discussion, that is the environment that I call the maker of phenotypes. And from caterpillars to humans, the environment is a main determinant of what we are. It's not in our genes, as they say. Your genes don't make you what you are. They are not in the driver's seat. They are just one element that is called upon when and during development. So we are going to see something that goes on in caterpillars. This was discovered in the 1880s, so that is, we know for a very long time these animals are exactly the same. So you take those animals and make them, you can put the eggs in the cold or in warm weather, and you will find exactly what I'm showing you that happens in nature. That is, at the left, the summer broods look like oak twigs, so they mimic that. But the ones that appear in spring mimic the flowers. So that is, the outside, the exterior world determines whether this caterpillar will look one way or another, and they are very different ways. But this doesn't only happen in caterpillars. In humans, it was discovered that children that were born to mothers that were severely malnourished during the Second World War, that is, during gestation they didn't have food, these kids, when they reach 50 years of age or so, they had cardiovascular diseases. And so this type of situation, and now there are many other cases being discovered, indicates that our phenotype is determined, in part at least, by the external environment, and then also by the internal environment. So here we have a diagram. This is the idea of that we were putting, people push for this idea with the molecular biology revolution, where everything was perceived as being controlled by genes, that development is a genetic program and the mother is just merely a fetal incubator, to what we know today, that is going back to 150 years and recognize the environment as the main determinant. So what we see today is that food, light, hormones, all these elements determine what you are going to be. And so your mother is your fetal environment and not just an incubator. So with this in mind, we can start looking at the problem of cancer or starting in the womb with a totally different perspective, not as a thing that you need a mutagen and it's in your genes and all that, but we can go to a different perspective that is called ecological developmental biology, where we acknowledge that there is no program or central control. Genes do not have a privileged causal role. The environment plays a main role in the determination of the phenotype, as I just showed you. There is redundancy and plasticity, and organogenesis is the result of reciprocal interactions among cells. So it's not that comes from the cells out, it comes from a dialogue among cells, among the society of cells. So how do organs form? I will give you an example, and you don't need to know everything here. What I'm trying to say is that 10 of the cell types that appear here in your kidney filtration apparatus are originated in this part that is like a pink thing in the upper part of the graph, the nephrogenic mesenchyme. And this is another part of the system that is called the Wolffian duct, and you don't need to know, but these arrows show that there is a reciprocal interaction among the two. So if you put in culture just one of those, the red or the green, you don't get anything. You have to put the two in order to get all this apparatus that filters your blood and so produces urine. And the influence of this green tube upon this red tissue is producing all these different cell types that you see here. So this is just to get into our minds the idea that this is very intricate, very interactive, and very complex, and if we touch it, you don't need to mutate anything. You can wreak havoc by just touching it. As I show you, the absence of thyroid hormone wrecks your brain, and if that happens during fetal development. Okay, so the next thing is how does this impinge in our notion of cancer. So Carlos Sonnenschein and I propose, after getting aware of what's going on with endocrine disruptors, we really started to investigate what was going on in the cancer field that didn't take into consideration the role of interactions among tissues in organogenesis, and we postulated the tissue organization field theory of carcinogenesis that is based on normal development. So here we have a tissue, like the mammary gland. Here we have the epithelium, are those cells with blue nuclei, and here we have the connective tissue that have cells that look a little bit like crescents. And as I showed in the previous slide, there are interactions between that the stroma here and the epithelium. If we put a carcinogen like bisphenol A in the system, those reciprocal interactions are disrupted, and as a consequence, you have abnormal development. And abnormal development means that the cells no longer know what to do in the sense of forming a given structure, and you have abnormal structures. And those abnormal structures, if the stimulus is maintained, will develop into cancer. So the tissue level is the level in which cancer takes place, and it's not a problem of cells getting crazy or just the cell getting mutated, but it's a problem of alterations of a dialogue between different cell types that form the normal tissue, and therefore, because this dialogue is interrupted, they become cancer. So we are now going to move into BPA, that is the chemicalized study. And as you can see, the same things that we have been seeing as the diethylestylbestrol syndrome appear here, and it's no surprise because BPA is a cousin, chemical cousin of diethylestylbestrol. Early puberty, as you know, is also a risk factor for breast cancer. And exposure in utero provokes early puberty, at least in the rodent models that we use. It produces altered mammary gland development, changes in the uterus and ovary, decreased fecundity and fertility, changes in the brain steroid receptor levels, changes in behavior, and predisposition to cancer and obesity. And you know that obesity is also a risk factor for breast cancer. So you see there are direct and indirect ways by which all the systemic insult by an estrogen at the wrong moment and in the wrong place that could occasion a very complex syndrome. And among those symptoms, if you will, is breast cancer. That is what occupies us today. Interestingly, there are some epidemiological studies that show that xenoestrogens could increase the risk of breast cancer. Well, first of all, we have to address the problem that the incidence of breast cancer has increased during the last 50, 60 years. And the hypothesis is that that is due to exposure to endocrine disruptors, because there are many endocrine disruptors that were introduced precisely after the Second World War. So that coincidence may be where we should look for to explain that epidemic. We know by epidemiological studies that gestational exposure to DDT increases the likelihood of developing breast cancer. And a very important series of studies done by Barbara Cohn in California show that this exposure has to occur either in utero or when you are a child in order to make a big difference in the incidence of cancer. In other words, exposure to DDT when you are 40 years old doesn't seem to be important in this case. It's whether or not you had it before, when you were a fetus or a child. Similarly, we know that diethylestylbestrol produced a very similar syndrome when people were exposed in utero. But there are therefore windows of susceptibility, and the fetal one seems to be extremely important. Experimental studies, like the ones we conducted, show that gestational exposure to environmentally relevant doses of bisphenol A increases the risk of developing mammary cancer. And why this concentration on bisphenol A? Well, because bisphenol A is a building block of polycarbonate plastics and a component of other plastics and epoxy resins, and it's prevalent in our daily lives, and is one of the highest volume chemicals produced worldwide. So we are all exposed, and CDC found that 92% of the urines they analyzed in a cross-section of the American population had bisphenol A. There are other chemicals too, but I'm concentrating on this one because we got to know what's going on by giving it to animals, a thing that we cannot do with humans, obviously. So this is what we found. This is very, very low doses of bisphenol A that are within the exposure levels in humans. So here we have a control. This is the mammary gland of an 18-day-old fetus of a mouse. And here we see many different tissues that are forming. This that I'm marking is the ductal system of the mammary gland. This is what is going to give all the ducts of the mammary gland. And this is set in this dark tissue that is going to become fat tissue later on. This blue thing that you see here is a component of the extracellular matrix called collagen. This is a control. Now we'll see the animal that has been exposed to bisphenol A just for a few days in utero. And you see that the blue thing, the collagen, has disappeared. If we look with more attention to this, again, I'm talking about this interaction between the fat tissue and the epithelium, etc., this interaction and conversation among tissues that determines the shape of the organ. You see that in the normal mammary gland, we see a lot of red stuff around this epithelium that is the ducts of the mammary gland. This disappears almost completely when the animals were exposed to bisphenol A. So you see very subtle things are going on here. But together with these subtle changes, you see that the stroma, that is the support tissue, matures very fast, forming fat earlier than in the controls. And that the surrounding connective tissue has an extracellular matrix that is altered and with a different composition. That in turn affects the epithelium. By the way, the receptors that perceive bisphenol A are here in the stroma, not in the epithelium at this age. So the changes in this stroma are inducing changes in the epithelium, that is the functional part of the organ. So we see here, this is the normal, non-exposed, this is the one exposed, and we see that it's already bigger. So there is an increase in the number of epithelial structures. But if it was everything bigger, it wouldn't be a problem. That is an acceleration of a whole normal phenomenon. At the same time that there is this increase, there is also a delay in lumen formation. That is, that these are cords of cells instead of being ducts. Ducts are like a pipe that has a hole, as expected by an organ that has to carry milk. So the lumen, that is the hollowness, is delayed. So we see things that go in both directions, and that creates a lack of communication that accentuates with time. So in order to study this and be able to see it in real time, we developed a method to culture the whole fetal mammary gland from the beginning, that is at day 13, to birth. And so what we see here is that the control, here is the epithelium, has this star-like shape. If it's exposed to bisphenol A, as you can see here, is much bigger. And this is at low dose of bisphenol A. If you put a truckload, it will totally inhibit the development. And this low dose, the environmental relevant dose, is the one that induces the propensity to cancer. This growth is inhibited by fulvestrant, which is an anti-estrogen that is given to women with breast cancer. So this is not for using in therapy in this case. What we are trying to do here in the fetal mammary gland is to see whether this is acting through the normal estrogen receptor that we know that is called estrogen receptor alpha, and the fact that it's inhibited by fulvestrant seems to indicate that that is the case. So you see that right there during fetal life we already see alterations that are measurable. What happens later? Well, here we are looking at animals that are 1 month old. That is the time of puberty in mice. So now you can see here this LN means lymph node, and these are the ducts of the mammary gland that are invading the stroma, the connective tissue. And you see that there is a lot of invasion. The invasion is carried out by these things that are called terminal embeds. But that is not relevant to you. The important thing is that look the distance traveled. When you give bisphenol A in utero, so here is already 1 month that bisphenol A is not in the system, you see a delay in this physiological invasion. Right there, the abnormalities persist even in the absence of bisphenol A. And you see already here, you see this kind of yellowish stuff. Those are cells that are dying. Those cells are dying in order to produce the lumen, to make from a cord of cells that is solid a duct that can transport milk. This is in the control, but in the animals exposed to bisphenol A, cells are not dying, so there is a delay in the formation of these tubes. Additionally, here we see a lot of nuclei that are stained sepia because they have this an antibody that detects progesterone receptor, which is very important for branching. And we see here a cluster of cells that are positive for progesterone receptor. That is where branching will happen. And we see that at 4 months of age, the controls are not very branched, but the ones that 4 months ago received bisphenol A are very branched and also have these little structures called alveoli. That is where milk is going to be produced. So we see that having these animals exposed during the fetal mammary gland development, that is day 11 to birth, that is day 20, then occasions a lot of perturbations that manifest later on during adult reproductive life. And here I'm going to show you something else. You see this interruption of the lumen, like the duct is blocked here and there. This is called intraductal hyperplasia. That is a precursor lesion to cancer. Cancers start with this type of lesions. If we look later on in proper, that was mouse, we can now use rats that are a better model for breast cancer. We see that those hyperplasias are increased at all doses of bisphenol A used with respect to the control by about 3-fold or more. If we look at those hyperplasias, not all of them are just hyperplasias. Some of them, like this one and this one, are carcinomas in situ. So bisphenol A increases the likelihood of getting carcinoma in situ. But if we will wait longer, we can also see palpable tumors. So we have one here that is small, and we see it in a whole mount. So that is the whole thing. And here is a histological section showing a cancer that is indistinguishable of those that we see in women. So here I would like to wrap up the why do we get there. And we get there, that is to cancer, because BPA acting in the fetus is affecting, is working through receptors of estrogens that are present in the mammary gland stroma. This altered stroma alters the formation of the epithelium. And here you see there is no mutation. It's simply aberrant morphogenesis. these two things produce a mammary gland that will have altered response to hormones. Like if you add estradiol in puberty, these glands respond much more intensively, and that is why you see that at 4 months of age they produce structures that normally are present during pregnancy and only in pregnancy. By BPA, this is directly acting on the mammary gland, altering the response of the mammary gland to hormones and altering its very structure. And then there is this other part that is bisphenol A also alters the hypothalamus. And the hypothalamus is controlling the ovarian cyclicity and therefore the secretion of mammotropic hormones like progesterone in the ovary, estrogens in the ovary, and prolactin in the hypophysis. All this together will give rise to an increased propensity to mammary cancer. And so if we wait long enough, this is what we are going to see. So to wrap up, my conclusions are that the fetal xenoestrogen syndrome challenges current reductionist paradigms and invites an integrative system outlook because there is all these interactions. And so we have to bring biophysics, that I didn't explain why, but we can discuss it, the study of cell-to-cell interactions, cell motility, and tissue dynamics to the forefront, because developmental genetics shouldn't be the only game in town. So it's not in our genes, it's in our development. It's in the formation of tissues where the problem is when you put a carcinogen, like in this case, a hormone mimic. The perinatal exposure to BPA triggers a florid syndrome, decreased reproductive success, altered behaviors, obesity, diabetes, cancer. And this is clear in in animals, and it will be very difficult to show in epidemiological studies because exposure is occurring in utero, and you have to see, and you will see the cancer at 50 years of age. But in animals, you can trace it perfectly well. So my question is it wise to keep exposing our offspring to these chemicals as they are developing? And of course, my answer is no. So here what we need is not treatment, is not best cures for cancer, is not this overrated precision medicine, is nothing like that. It's just not to be exposed. It should be a right not to be exposed. And I want to say that. I mean, if we were not exposed, we won't have this. Which brings us to the next issue, that is science and applied science. So one of the things that is going on is when we, the scientists that discover those things, pressure governments and agencies for regulation, the chemical industry comes and says, oh, but you don't really know all the mechanism. You don't know everything that is going on to produce that cancer. And I'm going to say, well, I mean, many years ago, when the cholera epidemics in London, that is considered the foundation of epidemiology, the founder of epidemiology, Snow, went to see where the cholera epidemic had its epicenter and say, well, it's this water pump that is creating the problem. So he disabled the pump and the epidemics ceased. So at that time, nobody knew what cholera was and that microbes were important for disease. So here we have to think that way, that if we have a problem like endocrine disruptors and we have animal studies that show increased propensity to cancer, we should start regulating these exposures. Basic science, that is the science of Newton and the science that we scientists like to do, is about living with uncertainty. There is always a new experiment to be done, a T to be crossed, an I to be dotted. 100 years elapsed between Galileo Galilei and Newton. Because, and this is not of essence, because really the orbit of the Earth didn't change in those 100 years. You see, this is about knowledge, not about action generated by knowledge. But when we are in medical practice, the outlook is different. I mean, if I'm in the emergency room, time of essence. As a physician, I had to reach conclusions and act now to prevent, cure, or save a life. That is a totally different way of using knowledge. When you do medical epidemiology, in order to know whether this chemical, this pharmacological agent is good for curing a disease, you have to choose the null hypothesis as the default, so you don't expect an effect, because it's best to err on the side of a false negative, so that your drug really has to do work in order to be perceived as such in your epidemiological study. But if you are doing public health epidemiology, you should do exactly the opposite. When studying exposures to potentially harmful agents, you have to choose the alternative hypothesis, that is that you expect a deleterious effect. And that is a sound practice, because it's best to err on the side of false positive. Then science, the basic science that is being done every day, will correct us. But meanwhile, in case that it was, our suspicion was true, we will have saved lives. And that is what we should be doing in this case and in any other case where public health is involved. So I want to acknowledge that this was funded, all these studies, by NIHS, and which is the only institute that cares in reality about the environment. And also I want to acknowledge the people that did the work, and this work was done in partnership with Carlos Sonnenschein. And what we saw today were experiments by a bunch of very talented PhDs, Lucia Speroni, Perina Swadia, Nicola Acevedo, Tessie Paulos, Mael Monteville, Cheryl Cheverly, Greg Witt, Michael Porrero, and Joe Traitman. And I won't mention the rest because of lack of time, but all of them were instrumental in this. I want to remind, among them, Laura Vandenberg and Marisa Maffini. Thank you for your attention, and I'm open to answering questions.
Dr. Soto, thank you so much. I open it up now to anyone who has a question, so please type it in. And Dr. Soto, I have one that has come through that said you noted that there was greater progress in Europe with regards to elimination of some of the chemicals of concern. Is there a correlation in the breast cancer rates in Europe versus the U.S.?
Well, it's pretty much the same. The progress was made 1 year ago, so as you know, breast cancer doesn't incubate in 1 year, so there is no changes right now. What is a big change in the way that things are being regulated. So now you cannot put bisphenol A in your food, and you cannot put phthalates in your food in France. And because when France says something, all the other, it's easier for the industry to use the same regulations for the whole European Union. And it's the same when California changes the standards, then the producers have to do the same for all United States. So the idea is the same. And a couple of months ago, I think it was, that the European Commission determined that bisphenol A is a chemical of great risk, and so it's put in a special list. So I think that they are doing more progress in spite of having the industry trying to, the lobbies are trying to slow down everything, like here. But I think that they are getting further than us, and they started much later.
And they started later than we did.
Yeah, because the whole thing, windspread, started here. And I think that they started, in 91 we had windspread, and they started to be active in 95. And but they have a more, government involvement in public health and all that. So there is another tradition. It's not like everyone is for himself like here. I mean, that is the general idea.
Okay, thank you. And do we have other questions coming through? So I do have a follow-up that's come in, which I would be interested in hearing your thoughts on. It's really the question of, so how do we get other government agencies to pay attention? You said that we're not getting the same level of attention here in the U.S. And so
activism.
Yes?
I think that we were very good when, you know, in the early 90s, the Breast Cancer Coalition and all these people started pounding their doors. We have to continue, but somehow there was a change. I mean, I think there was a change where people saw the idea that more research is needed instead of more action is needed. And I applaud some of the breast cancer activists that are working just telling producers of bad things to stop and things like that. But I think that the government should be a very important target. I know that the public is doing something, like, for example, people are checking for makeup, for example, and that doesn't contain as many toxicants, and there are some companies that are trying to do that. I know that there are companies that are trying to change the lining of the food cans not to shed bisphenol A, and etc. But I think that it's, you know, you have to pound the doors of Congress and work very, very, very decisively in order to get regulation. That is what they have been doing in Europe, because the agencies didn't pay any attention because they are full of lobbyists pounding at their doors. So they just went to the Congress. That is the case of France. The activists went to Congress, and there are some physicians in Congress, and then the law was passed unanimously in the House and in the Senate.
Great, thank you. Let's see if we have any more questions. Well, Dr. Soto, I have to say, on behalf of Cheryl Osimo, our Executive Director of the Massachusetts Breast Cancer Coalition, and the entire Board of Directors, I want to thank you for sharing your knowledge with us today. I would also like to thank all of you who are listening for joining in today, and I wish you all a great afternoon. Thank you so much.
Thank you very much, Michelle and Cheryl. Bye now. And
thank you.
Bye-bye.